Good afternoon. Welcome to Medical Grand Rounds. I'm Bob Wachter, Chair of the Department of Medicine. Welcome to our small and mighty group here and 50 to 100 people who are watching online currently. We will continue doing Grand Rounds through the month of June, then we're taking the summer off. July and August restart after Labor Day, unless there's some big breaking news, more likely COVID than anything else, in which case we'll schedule a special session. Closed captioning is available. If you have Q&A, please type them into the box for those of you that are watching virtually. So we have an exciting session today. My core belief is that medicine is undergoing a revolution, a pretty slow revolution, one that will take the next 10 or 15 years to play out. But the revolution really began 10 or 15 years ago when all of our healthcare data began to be stored in digital form. And it is accelerating as we begin to take advantage of that digital data to answer important questions and interact with patients in very, very different ways. And I think that revolution is going to transform healthcare in all sorts of ways, some predictable and some completely unpredictable. And so the topic today is the science between real-world data, answering today's questions today, which really gets at part of the heart of the question of how do we use all of this data that we're collecting to make better decisions, and both as clinicians and also as patients, as they increasingly will have control of the data themselves. For that, we have four terrific speakers. The format will be four short lectures and then a Q&A session or discussion session at the end. I will briefly introduce all of them, and then we'll have one after another come up. One is Uma Mahadevan, who is professor of medicine in our division of gastroenterology at UCSF Health. Uma trained both here at UCSF and a fellowship in inflammatory bowel disease at Mayo. And she is director of the UCSF Colitis and Crohn's Disease Center and directs our IBD fellowship. She has earned a well-deserved national and international reputation as a leader in the field of inflammatory bowel disease, which increasingly is taking advantage of data to make better decisions. Next will be Marina Sirota, who is assistant professor in the Baker Computational Health Sciences Institute here. Prior to that, she was the lead research scientist in the Division of Systems Medicine at Stanford and also worked at Pfizer. Marina completed her PhD in biomedical informatics at Pfizer, and her work involves leveraging and integrating different types of data to better understand the role of the immune system in disease. Seems quite topical these days. She was awarded the Young Investigator Award from the American Medical Informatics Association in 2017, and also really emerging as national leader in her field. The next is Erica Ferran, who's assistant professor in our division of pulmonary critical care, sleep, allergy at UCSF Health. She attends the interstitial lung disease clinic and on the pulmonary consult service, and she conducts interdisciplinary research focused on improving the healthcare safety quality of patients with interstitial lung disease. And toward that end, she has also been using real-world data to come up with insights. And last but not least, we have Atul Butte, who's the Priscilla Chan and Mark Zuckerberg Distinguished Professor and Director of the Baker Computational Health Sciences Institute at UCSF. Atul also is the chief data scientist for the UC Health System, which is enormous and provides him and the people he helps, whose work he facilitates, access to one of the most robust clinical data collections in the United States. Atul's had an extraordinary career as an academician with hundreds of published articles and continuous funding from federal agencies and foundations. He's also quoted everywhere appropriately, and he's also had multiple patents and co-founded 3 companies. So a really interesting and a diverse career, making a tremendous impact in data science everywhere around the world and around the country. And we're particularly grateful that he's here at UCSF, where he helps lead all of our efforts. So with that, I will turn it over first to Uma.
Hey, thank you, Bob. It's a pleasure to be here. So let's start first with some definitions. What is real-world evidence? So the 21st Century Cures Act defined it as data regarding the usage, benefits, or risks of a drug from sources other than the traditional clinical trials you use to get drugs approved. The FDA expanded this to any medical product and data derived from analysis of real-world data. So your next question is, what is real-world data? It's data relating to patient health status and delivery of healthcare collected from a variety of sources. This can be randomized trials, pragmatic trials, observational studies, data mining the electronic health record, claims databases, patient-generated data from devices, and many other settings. So there's a lot of real-world data that we study at the Inflammatory Bowel Disease Center. Some examples, a pragmatic trial is MOSAIC, which looks at inpatient versus outpatient IV steroids for patients with severe colitis. If you're just in the hospital to get your IV steroids every day, is that really necessary? The IBD chat is a collaboration with CDHI. It's a chatbot that we send out to our 3,000 patients with IBD we see per year, and they enter their symptoms, their data, their medications, and if they have a red flag alert, it goes directly to our Epic inbox where we're able to communicate with them and say, hey, how you doing? But also what it does is it collects prospective data in ways that are collected in clinical trials. So we can go back and do real world evidence data on drug exposure and benefits using the chat. We use the electronic health record in many ways, including trying to mimic clinical trial outcomes. We worked with Encore to develop a Tableau dashboard for our IBD patients, so we can, for example, look and see how many patients we gave off-label upadacitinib to and how did they do, and that was recently submitted for publication. And then also across the state, there's a California IBD cohort led by Sid Singh at UCSD, which has 30,000 IBD patients across 7 medical centers on immune-mediated drugs that we can study and assess for outcomes. But my area in particular is pregnancy. And pregnancy in particular is a very challenging area to do drug studies. There is a paradox where in order to protect the fetus, drugs are often withdrawn. And what happens is the mom has a flare of her disease and the fetus does worse. So that is not actually the best outcome, but the default quite often is to withdraw therapy from pregnant women. But this goes back many, many years to all women. In 1977, the FDA policy excluded women from clinical trials, even if they have low or no likelihood of getting pregnant. In 1986, the NIH policy encouraged researchers to include women, but it was poorly communicated and not really carried out. It was not until the first woman head of the NIH, Dr. Bernadine Healy, that it became law that women and minorities should be the subject of clinical trials. Another issue has been the FDA labeling. You may remember back to the A, B, C, D, and X labeling, which we no longer use as of June 2015. We now have the PLLR, which gives you data from which you can make your own decisions. The A, B, C, D, and X was again used to deny women care. When I first joined the faculty at UCSF, there are many obstetricians who refused to care for my patients if they continued their thiopurines. And so hopefully we have moved beyond that. But have we really? So what happens today? Women who become pregnant must drop out of a clinical trial, even if they're on a drug with known safety benefits. For example, anti-TNF agents. There are 4 or 5 or 6 anti-TNF agents. You get pregnant in a trial, though we have thousands of patients' safety data, you have to drop out. In clinical practice, rheumatologists and even gastroenterologists may deny women of childbearing age the use of a JAK inhibitor because they're afraid they may get pregnant and animal data has shown some risk. And then medications with well-established safety profiles over the last 20 years, such as anti-tumor necrosis factor agents, are stopped early in pregnancy. 68% of women in one study who stopped their TNF did it on the advice of their rheumatologist. And a rheumatologist will tell you, they get better in pregnancy, but that's actually an old doctor's tale. There is absolutely zero high quality, medium quality, or even low quality data to support that. And women with rheumatoid arthritis have significant adverse outcomes of pregnancy related to inflammation, such as preterm birth, preeclampsia, et cetera. And then finally, the European Medical Association, which is the equivalent of the FDA in Europe, sent a letter in March of 2022, this year, that said infants exposed to infliximab in utero should not get a live vaccine for 12 months. Now, we know that monoclonal antibodies are actively transported across the placenta, but by 6 months, the levels are either gone or negligible. So to deny BCG vaccine in Europe, where the number one leading cause of infectious mortality in children is TB, is certainly harmful, with zero benefit from denying vaccine. What's even worse is they said if a mother is breastfeeding on infliximab, regardless of how old the infant is, so you could have a 14-month-old, the child should not get live vaccine. So that goes to MMR and varicella as well as BCG, based on zero data showing harm and pretty good data showing no risk. So for pregnancy and inflammatory bowel disease, there are a lot of questions. What is the safety of biologics such as infliximab and small molecules such as tofacitinib, upadacitinib in pregnancy and lactation? We know that small molecules can cross in the first trimester, but biologics should not cross in the first trimester. They are monoclonal antibodies, and their FC portion becomes actively transferred across the placenta beginning around week 14, with 80% of that transfer occurring in the third trimester. So what does that transfer mean for the infant, for the risk of infection, developmental milestones, vaccines, and even daycare? So in 2007, we launched the PIANO registry, which is Pregnancy, IBD, and Neonatal Outcomes. This is a prospective national registry, including 30 sites of the Crohn's Colitis Foundation Clinical Alliance. To be enrolled, you had to be pregnant with IBD. Initially, we followed them out to 1 year, then 4 years, and now we're following them out to 18 years. There are about 1,600 women enrolled in this registry, and they're divided into 4 groups. No immunosuppressant biologics, azathioprine 6-MP, biologics or combination with azathioprine, and then small molecules. And in 2021, we published our main results, which looked at 1,490 women who had 1,431 live births, 379 not on immunosuppressants biologics, 242 on thiopurines, 642 on biologics, primarily anti-TNFs, plus an additional 227 on a biologic plus a thiopurine. So 900 prospectively followed biologic patients. There was no increase in congenital malformations, spontaneous abortion, preterm birth, low birth weight, or infections out to 1 year of life. But if they had preterm birth, which was associated with disease activity, there were increased infections. They had active disease, there were increased spontaneous abortions. So the biggest risk to the pregnancy was active disease in the mother. The use of any of these medications did not impair developmental milestones using the Ages and Stages questionnaire out to 1 year. Now, physicians will say, or providers will say, well, I'll stop the biologic, and if they flare, I'll give them steroids. How do steroids do in pregnancy? Increased risk of preterm birth, small for gestational age, low birth weight, and NICU stay at birth. And use in the first trimester was associated with an increase in birth defects. So you're not helping anyone by taking them off a biologic, which has shown safety, and switching them to corticosteroids. Infections at 4 months and at 1 year were not increased by drug of exposure, whether it's biologic or thiopurine or both. And then what about breastfeeding? So we looked at levels. So we know that when it crosses the placenta, as with all antibodies, the babies are born with higher levels of that antibody than the mother. And for example, for infliximab, the infant can have 2 to 3-fold higher rates of infliximab at birth than the mother, which is why we don't give live vaccines for the first 6 months of life. But for breast milk, the amount of transfer at peak, so we measure after giving the drug and then measure over the next 7 to 14 days, the peak is 0.74 micrograms per ml, and that's maybe for a day or two, which is not something that's going to impact vaccine harm. And breastfeeding while on biologics did not adversely affect infant growth, developmental milestones, or infection rate. Despite this and other supporting data, the EMA still came out with their default of not giving vaccines if the infant is breastfeeding and the mom is on a biologic. What about response to vaccines? Maternal immunomodulator or biologic use did not impact vaccine response. It was the same whether they were given a biologic or not. And even though we tell them not to get a live vaccine in the first 6 months of life, we had 40 patients who received rotavirus vaccine, and they all did fine, even if they had significant levels of infliximab. And this is similar to the JAMA paper where varicella or shingles live vaccine were given to older rheumatoid arthritis patients, and again, they were fine. Now, my favorite is daycare. So in Europe, they recommended that if the mother was on a biologic during pregnancy, the infant shouldn't go to daycare for a year. Great if you're in Europe. In the US, when you get 6 weeks, that's just not feasible. So we looked at that in our piano registry as well. And we found that if you went to daycare, you had a higher risk of infection than if you didn't, but whether you were exposed to a biologic or not did not make a difference. So to summarize my section, real-world evidence provides guidance for questions in populations that are underrepresented or not represented in traditional clinical trials. Drug safety in pregnancy requires real-world data and real-world evidence. And the challenge is that safety data is never enough. So when you had 1 year of data, they're like, we need more. 4 years of data, we need more. 18 years of data, hopefully we'll get there. But it requires engaged investigators and patients over a sustained period of time, and raising funding free of pharmaceutical support, which I think is important for drug trials in pregnant women, can also be challenging. Thank you.
Hello, thank you so much for having me. And I'd like to tell you a little bit today about our work in the context of using real-world evidence data for drug discovery, shifting gears a little bit. So there's an amazing opportunity to leverage different kinds of data for drug discovery, and specifically drug repurposing. It takes a long time and it's very expensive to take a drug and bring it to market. And there's lots of data out there. There are molecular data in the public domain that we can leverage. People have been using different types of omics technologies in creative ways in the last few years. And finally, there's data from the electronic medical record, or the real world data that we can use to validate some of these hypotheses. I come from the background of using molecular data. So actually, during my graduate training with Dr. Butte, who you will hear from later today, we developed a computational pipeline for drug repurposing, or identifying new uses for existing drugs, using gene expression data. And the idea is fairly simple. We can take a disease expression signature, query it against the drug data, and identify drugs that might be reversing that expression signature back to normal, with the hypothesis that if that reversal is there, then maybe that drug could be used as a potential therapeutic. We have recently applied this approach in collaboration with Dr. Yidong Huang at the Gladstone Institute to look at Alzheimer's disease. And here we really took a precision medicine approach. We wanted to stratify the Alzheimer's disease patients by their ApoE genotype and see if we can identify compounds that might be specific for individuals with ApoE44 disease. So again, we started with public data. We identified individuals who stratified them by their genotype and identified signatures specific for each genotype: ApoE33, ApoE34, and ApoE44 disease. Looking at the dimensionality reduction plots there, you can see that there's a separation by case control, but also a separation by genotype when we look at these individuals. When we look at specific genes and pathways, we see that some of the signal is shared, but at the same time, there were some signals that were specific for each of these groups of patients. So then how does that translate to drug discovery? We queried the expression signature against the drug data that we had access to, and saw that there was a specific drug called bumetanide that specifically reversed the ApoE44 signature, shown here on the top left-hand side. However, the signals for that drug across the other signatures was not as strong. So there was potentially still therapeutic potential, but not as strong as what we've seen for ApoE44 disease. And that is visualized here. So bumetanide happens to be a loop diuretic that works by decreasing reabsorption of sodium by the kidneys. It's used to treat edema and hypertension. The animal work in Dr. Huang's lab has shown that treatment with bumetanide rescued AD-like cognitive and electrophysiological deficits in ApoE4 knock-in mice. And we were also able to leverage single nucleus sequencing, so looking at this data on a single cell level to explore post-treatment effects in mice as well as in human cells. But the question that we're going to focus on for the purposes of today is, can we further, or how can we further validate such hypotheses or such findings with real-world data? So we have access to electronic medical records data from UCSF. This data spans from 2012 until today, and the data that we were using at the time spanned about 1.3 million patients. So the electronic medical record contains data including the diagnosis codes, encounters, lab test results, medication orders, procedures, pretty much all the information that's captured when a patient sees a clinician. To give you a snapshot, this is what the demographic information looks like. So the age distribution across the patients at UCSF, as well as the ethnic background, as well as information on sex that we have on the individuals. So to test the hypothesis that bumetanide might be associated with reduced risk of Alzheimer's disease, we started a case control study. So and this was work led by Edith Kosti in my lab in collaboration with Tomiko Oskotsky here at UCSF, but also we did an independent validation at Mount Sinai in collaboration with Benjamin Glicksberg and Mesut Bijak. So at UCSF, we started out with 1.3 million patients. We identified 5,500 roughly individuals who've been exposed to bumetanide. For about 4,500 of those patients, we had complete demographic data, and we carried out 1 to 2 case control matching using propensity scores matched on age, sex, race, and ethnicity, as well as the indication for bumetanide, which is hypertension and edema. And we wanted to see what was the prevalence of Alzheimer's disease in both groups, individuals who've been exposed to bumetanide versus not. And we saw that looking at the individuals who have been on bumetanide, we saw a significant lower prevalence of Alzheimer's disease in that population. This has been replicated in an independent health system at Mount Sinai, where we started with about almost 4 million patients, identified about 3,000 individuals who've been exposed to bumetanide, and we also filter by age, so individuals over the age of 65. Here, we saw a consistent signal, even a higher difference. And this was our first attempt at using electronic medical record data to try and validate a therapeutic hypothesis. We've expanded on this work since, and especially during the pandemic, there were several hypotheses that we were interested in testing in the context of COVID-19. And one resource that we were able to get access to, which is an amazing effort by Cerner to aggregate information on patients relating to specifically COVID-19. So what they've done is they've aggregated emergency and hospitalized patient diagnosis, about 500,000 patients who've been diagnosed with COVID-19 across 87 institutions in the US. The split is roughly equal between males and females. And in terms of the data that was made available to researchers includes demographics, medications, procedures, encounters, diagnosis, lab test results, as well as medical history for up to 5 years. So there were several hypotheses that we were interested in looking into. The first one was looking at role of statins in the context of COVID-19. Statins are used primarily for lipid reduction, and they have known anti-inflammatory and immunomodulatory properties. And in patients with COVID-19, retrospective studies have shown that they're associated with reduced disease severity and disease mortality rate. And this is what we wanted to investigate further. This is work in collaboration with Dr. David Stevenson at Stanford and was led by Ivana Marek and Tomiko Oskotsky. So we used a similar model. We identified a number of patients who were exposed to statins from the Cerner database at the time, and carried out 1 to 2 matching with controls who were not exposed. And we were able to see that consistently across 10 different iterations of this analysis, there was a reduced mortality rate in individuals who were exposed to statins. We did a very similar analysis looking at mortality risk in COVID-19 patients who've been prescribed SSRIs. Following a very similar analysis procedure, we started out with about, as I mentioned, 500,000 patients who've been diagnosed with COVID-19. About 3,500 of them have been exposed to SSRIs during a defined time window. And here we looked at specific subtypes of SSRIs. We looked at fluoxetine or fluoxamine, as well as other SSRIs and matched controls. So we queried the Cerner data, and what we were able to observe is a reduction of about 8% in relative risk of mortality among patients with COVID-19 who've been prescribed to SSRIs in comparison with the matched control cohort. We saw a much larger 28% reduction in mortality rates, specifically in patients who were treated with fluoxetine, and 26% reduction in patients who were treated with either fluoxetine or fluoxamine. So why SSRIs? There has been a link between serotonin and the immune system, and we've seen that there's potentially a decreased levels of certain cytokines, such as IL-6, in patients treated with SSRIs. They might modulate sigma-1 receptor pathway, thereby reducing damaging effects of inflammatory response, or by inhibiting the acid sphingomyelinase ceramide system. However, these are just hypotheses. The important thing to note is that we can't get at this just gives us association. We can't get at the mechanism of action or any type of directionality from this type of simple analysis, but at least it gets us a little bit closer to leveraging human data to validate some interesting hypotheses that we might have. So in terms of ongoing work, we're developing and applying computational approaches, leveraging both molecular data as well as real-world data for drug discovery. It's an amazing opportunity to use this type of data for these what I call in silico clinical trials. But of course, there are many challenges as well. For COVID-19, there are clear outcomes that we can look at, such as mortality rate or hospitalization. However, for many more complex conditions, this is much, much more difficult. There's many missing data, and there are challenges in aggregating these data across institutions. However, again, this is a way to start the discussion of how we can integrate both clinical and molecular data to hopefully get us a little bit closer to precision medicine. This is the team, both my group here on the left-hand side, as well as our collaborators at the Gladstone for the Alzheimer's disease work and at Mount Sinai and Stanford for COVID-19 work, as well as our funding sources. Thank you.
Good afternoon, everyone. My name is Erica Ferrand. I am co-director of the UCSF Interstitial Lung Disease Program, and my research combines data science, digital health, and implementation science to try to advance the delivery of high-quality, safe, and effective care in ILD. And I'm really excited to talk about how we are leveraging real-world data sources to advance those aims. I will say that we are really early in this process about thinking about ways to use this type of research to advance care in ILD. And yet, because it's a rare disease, real-world data has the potential to be a real powerful driver of a research platform. And so I'm going to give two examples of how we have used it to answer really fundamental questions in our field. Although this is the story of how we are using real-world data in interstitial lung disease, it is not a presentation about ILD. In fact, the majority of what I'm going to share is widely applicable to other disease models. But there are a couple of things about ILD that I do think it's important that you know. The first is that it's rare and it's complex. There are over 200 subtypes of ILD, which can make diagnosis and management decisions really difficult. But there are a lot of similar features that we can leverage when we're thinking about doing real-world data research. That is common symptomatology, common diagnostic approaches, and then most importantly, these are high healthcare utilizers with a high burden of comorbidities. And because of that, they are really active in a health system and generate what turns out to be a very unique digital footprint in the EHR. I can tell you as a health services researcher that there are many unanswered questions in ILD, and I am particularly interested in questions related to how we approach management and integrating evidence-based practices across the continuum of a patient's natural history of disease, and what tools we need to be pulling in to be able to execute that. But there are many barriers to doing clinical research in a rare disease. Particularly, it's resource intensive, and we also have trouble meeting enrollment targets because it is a rare disease. And so real world data offers an opportunity to address some of those barriers. Although interestingly, electronic health record data, which is probably our most accessible data source of real world data, has really been underutilized. And so I want to show examples of where we found that to be really helpful. So the first challenge that we looked at was cohort identification. So identifying rare disease cohorts is a time-intensive, labor-intensive, and costly first step, but it's also essential. The way this is accomplished right now in ILD is largely manually, right? A patient comes to a healthcare system, they undergo an evaluation, they're diagnosed with ILD, and if it's a specialty care center, that information is captured in a database. This plays out nationally with a subset of patients being referred to and enrolled in a national database that's run by the Pulmonary Fibrosis Foundation. And so these are well-curated registries of information, but they are resource dependent, expertise dependent, and for that reason are really difficult to scale. Alternatively, you could think about developing an algorithm that you could deploy in a health system and automatically identify patients with interstitial lung disease, right? A much easier and less laborious approach to identifying patients. And the benefits to that are many, but there's the potential for generating more representative data sets. Now we're identifying patients that are excluded for or underrepresented in registries. By working with different types of data, we could better estimate the national prevalence of ILD, which is important for policy implications. But the kicker is that those algorithms run almost exclusively on ICD diagnostic codes, which turn out to be really poor predictors of ILD. And so the algorithms that are built on them have really performed modestly and have not had really robust uptake. So we asked, given what we know about patients with ILD, that they have this big digital health footprint, and that we have way more data in the EHR beyond diagnostic codes, can we leverage high dimensional medical data to automate ILD cohort development? And to do this, we started by thinking about how is data at UCSF captured, organized, and stored, and then identified the de-identified clinical data warehouse as the place where we wanted to work, which has information on over 5.5 million patients and includes both structured and unstructured data, which is an unbelievable resource, and pair that with our well-labeled data set from the UCSF ILD database, and having that is really important if you're going to train an algorithm. You need some ground truth. We took all of that patient data, and then we used clinical domain expertise to say, what are the features that we would want an algorithm to consider? So again, this is a structured approach to algorithm development. And we selected a number of different classes of features, including demographics, code counts, which included diagnoses, procedures, medications, encounter visits, lab data, particularly pulmonary function tests and CT scans, things that we know to be important in the diagnosis of ILD, and then also concepts from clinical notes, since we had this availability to extract data from clinical notes. We used a feature selection process to then, out of those candidate features, identify the ones that were most predictive of ILD, and then trained an algorithm using a gradient boosted tree classifier. This work was done with colleagues at the Baker Institute. And the result was that we trained not one, but 3 machine learning algorithms that reliably identify patients with ILD within a general adult cohort of EHR patients. For those who are not data scientists and are listening, myself included, precision is positive predictive value, recall is sensitivity, the F1 score and average precision you can think of as sort of composite measures of those two. And what I'm showing you here on the table is that in both training and testing, our best performing model outperformed significantly anything that has been developed or published thus far in ILD. Not only that, but we thought about the real value of an algorithm is not just running it in a center like UCSF, but being able to deploy it in other EHRs. And so we have to think about what data is available in other EHRs. So while our best performing algorithm used all features, including unstructured data, we intentionally trained an algorithm that did not use unstructured data and excluded pulmonary function tests, a variable that's known to have a lot of missingness in many EHRs. And all of them performed quite well. The second challenge I want to tell you about is how do we address evidence practice gaps? So as a health services delivery researcher, I'm really interested in, once I've identified a cohort and I know nationally that there are evidence practice gaps, how do those evidence practice gaps play out in my local health system? And then can I develop an intervention, implement it, and study it? And I'm particularly interested in digital interventions that addresses that practice gap, and then look at the impact and outcomes. In order to do that, I really need a comprehensive system to be able to conduct that kind of research. And yet, what I actually have are two parallel systems, right? I have registry data, which is really structured to support population health research with consistent data definitions and uniform data collection. And then I have the EHR, where I do the work of patient care and patient management, and where I would potentially implement interventions to address these evidence practice gaps. And so what we asked was, is there a way that we can begin to modify the EHR so that data collection more closely mirrors what happens in a registry or a clinical trial, and then be able to harness that so we can use the EHR for expanded secondary use, namely research? To do that, we had to start by doing a baseline assessment of what ILD research or what ILD data looked like in the EHR. And I'm not going to lie, the current state was scary, right? There was a lot of data missingness. There's a lot of data redundancy, data points that are captured in multiple different data fields and conflict. Mission critical data like ILD diagnosis was not easily extractable, and as I already told you, is not well represented by ICD codes. But the good news was that almost all of the information we wanted was there. It just wasn't accessible, and it wasn't reliable enough to use for a secondary purpose of research. And so we have been working with the Apex-enabled research team to say we need a single truth of data, ILD data in the EHR that can be used for both clinical and research purposes. And how can we develop tools to be able to do that? So this is the first of many tools that we are developing and we are currently piloting, which collects that information, that mission critical data like diagnosis, standardizes the collection of process measures, and then begins to build in other features that support providers so that they can use these tools during their clinical workflow. So now that I've told you a little bit about where we are using real world data in ILD, I'm really excited about telling you, where does that allow us to go? So by automating cohort identification, we are now able to ask, how does variability in access, use, and delivery of ILD specialty care impact outcomes? Both at UCSF, looking at patients who have ILD and receive specialty care versus those that do not. We are positioning ourselves to use UC-wide data to ask this question across our health institutions, and then really thinking about how we can partner with our colleagues at Kaiser to look at a non-specialty care center and see how this is different. By improving secondary use of EHR data, we're now asking, how can we improve provider adoption of evidence-based practice guidelines and developing clinical decision to support tools that we can deploy in Apex and then be able to study its impact in Apex. And then I haven't told you about sort of where we're going with integrating new data streams, but just really quickly, because we're in the era of telehealth and interested in kind of collecting information about patients when they're not actually physically with us, we are doing a pilot of using remote patient monitoring, which collects patient oximetry data and spirometry data. And that is positioning us to be able to ask, how can we improve patient self-management in ILD? So this work was possible due to support from an absolutely fabulous mentorship team, collaborators, unparalleled institutional resources at UCSF, and generous funding and support from a number of sources. And then a special shout out to my first year fellow class, who are all assistant professors here and are doing fantastic work in pulmonary and critical care, and who supported me when I was a first year fellow with nothing more than a vision board and nonsensical rambling about the promise of big data. I have given you a really quick intro into the why, the what, and the how of what we're doing, and I really invite additional conversation offline.
Great. I get the back clean up here. So my name is Atul Butte. I'm the director of the Baker Institute and the chief data scientist for the UC Health System. So you just heard three great speakers, and I'm going to try to present more on how you can actually do what these three speakers have shown you what they can do. First, the kind of general premise, I think when we talk about real world data and real world evidence, we're often now really meaning electronic health record data. And so the way I usually start a talk like this is really to build on this premise that we have spent collectively as a country hundreds of billions of dollars putting in these electronic health record systems. So to me, the narrative is it will be a national tragedy if we don't use this data to improve the practice of medicine. Having spent hundreds of billions of dollars here, right? You see three press releases here from including some of our neighbors here in California. Of course, we have to do this safely, responsibly, respectfully, but what a tragedy it will be if we don't all learn how to use this data to improve what we do. Given that I think now this is the most expensive data in America. We're paying doctors to type all of this in. Now, a lot of the kind of impetus for me to do this really comes from the FDA. I'm an NIH-funded person. I've been funded by NIH for a long time. But more and more of my kind of motivation comes from the FDA, not NIH. And obviously, they've been putting out guidance like this, really showing us as academics, but also pharma, biotechs, device makers, the importance of real-world data in getting their jobs done and getting products approved. The three classic examples, there's a couple shown here. I want to make sure people get, because it's a grand rounds. The top left one in the back there is approval of Ibrance for male breast cancer. Now, male breast cancer is certainly not a common disorder. It would take a long time to collect all the males with breast cancer to run a prospective clinical trial. But so many patients were being treated off-label with a drug for female breast cancer, and that data was collected in electronic health record data, that Pfizer was able to then gain approval, right, a label approval for Ibrance for male breast cancer solely using clinical data, right? Not having to run a clinical trial. So that really opened the floodgates, saving them hundreds of millions of dollars, getting a drug faster to patients. And now it's already been shown in heart valves and in the device world, and the FDA really accepts this. Now, on the right side is an even newer concept of single control arms or synthetic control arms. It's not synthetic data, but where the drugs now might be tested in one cohort and the control group might be somewhere totally different because of just how we can use electronic health record data. And we at UCSF are entering into synthetic control arm types of studies now as well. So FDA gets this, pharma and biotechs get it. That's why it's really an exciting time. Now, how would you get it, and how would you be able to do any of this stuff? Information Commons, you see the URL at the bottom, that we have created, we meaning the Baker Institute, but with a whole host of collaborators, including the CTSI, CDHI, SomTech, the UCSF Library for Education, and many, many others, putting all the data, tools, compute, and community all in one place, already de-identified, and no further IRB approvals needed to use this data. This is multi-factor data, so you got now just looking at UCSF, up to 4 million patients, 250 million diagnoses, 71 million medication orders. You see all the rest of the numbers here, including 115 million clinical notes that have been de-identified. I think that's really the fun part of what we've been able to do. You see 600 people already onboarded using Information Commons. The notes, as you've heard about from some of the previous speakers, 115 million notes, but we've already extracted 5 billion concepts, auto reading or computer reading some of these notes to try to figure out what we actually put in those notes. If you don't believe what the computer is reading, you can read them yourself. They're already safely de-identified and accessible. We're very excited to work with radiology to get the radiology images in there. And then again, de-identified but linked based on that same synthetic patient ID. Nearing 8 million DICOM files or DICOM file images of all sorts of different modalities available. This is real. I'm not just kind of blowing smoke here. Here's some quotes, maybe from some in the audience here. We save these quotes because we use it to kind of sell it to others here at UCSF. That I think Information Commons is there for you. This is there. It's not just magic. And we really want to make sure everyone, especially the Department of Medicine, knows that they can access this data and actually use it to thrive. And just some headlines of some of the papers, especially COVID-related papers that came out over the past year or two. And in fact, I'm just going to put a plug for one of our papers where we were able to use rheumatoid arthritis data, not just patients at UCSF, but also at San Francisco General, and use 2 different data sets to compare to predict the future, for example, training in artificial intelligence to predict the next 90 days for a patient. So we got many of these, and we're enabling many more, and that's important. Now, we also know pharma and biotechs want to do this with us, but we have to do this in a safe, responsible way. So the Baker Institute's launched a Center for Real World Evidence here. It's just evidence.ucsf.edu. And that's where we're starting to collect a lot of these pharmaceutical types of studies. We don't give them the data. We take in a question. If it's interesting to us too, then we jointly answer that. But we'll find a co-PI in many of the departments in medicine, surgery, and all the rest who are also interested in asking those questions. So we might shepherd inbound those types of agreements and get those done. Vivek Reddy wrote a paper about 2 years ago. It's been cited dozens and dozens of times on 21 uses for real world evidence. Vivek's in the GI division here in the Department of Medicine. And the dots here indicate how many pharma contracts we've already been able to sign. Again, safely, responsibly doing this type of work. We don't have to give out the data. We're just figuring out answers together. The yellows mean ones that are actually in discussions or in contracting. The greens are ones that have actually already been executed. So I think we got a real model here for safely doing these types of studies at UCSF, and we're going to keep helping junior faculty thrive by bringing them those projects. Now, in the last few minutes, I'm going to take off my UCSF hat, put on my UC-wide hat, because everything you just heard about at UCSF, we can now start to do across the entire University of California. This is the kind of logo diagram you see with our sibling institutions. Of course, we have UCSF, but UCLA, Irvine, Davis, San Diego, and Riverside. Riverside is tiny. They got a couple of thousand patients, right? No hospital there yet. The other 5 obviously are massive. And every month, all of that data flows centrally into a central data warehouse that we can use for operational improvement, quality care, but of course, research as well. This is a busy slide, but with the latest numbers, so 8 million patients, right? The UCSF and UCLA installed Epic more than 10 years ago. So now we're coming up on 10 years of longitudinal data on some folks. 310 million encounters, 875 million procedures, and my favorite, 1.1 billion medication orders or prescriptions we've written on these patients. Or as I like to say, everything from Tylenol to CAR T cells. Tylenol or acetaminophen is probably the cheapest thing we can give to a patient in the University of California system, and CAR T cells is probably the most expensive thing we can do to take out their immune fighting cells, train it to fight cancer, and then put them back. And we got everything in between, merged with California regulatory data, some text data again, text comes up. The death index for California is already intersected here, and we do this constantly harmonizing all the new drugs that come out every single week. To kind of give you an idea of this, here's just pick one lab test, lymphocyte counts. Here's 14.2 million of them, right? We can do this for any blood test we have, right? From 0 to 100, I guess this is cells per 100 cells. I don't even know what kind of distribution this is mathematically, but it looked interesting to me. We could do this for even the genes in the cancers that we're sequencing and all the rest here. We got in a central data warehouse. Now, close it out. Why is this impressive? Why are pharma companies trying to really respect what we have here? Here are just 3 well-known, well-used drugs at UC and UCSF. And just to give you an example, I'll just take the first one. Humira is a great biologic that we use like crazy out of AbbVie. And it's approved for rheumatoid arthritis and inflammatory bowel disease and many other indications. But if you look at the original pivotal studies, which you can get off their website, you see a couple hundred patients, right? Maybe 500 patients, 600 patients. And in those studies, not all of those patients were even treated with the drug, right? There's control arms there too. And then once it's approved, boom, We've treated 11,000 patients with Humira now at this point. So at a certain point, you could see the similar numbers. You could see those kind of numbers for everything in medicine. At a certain point, once a drug or device is approved, we use it like crazy at UC. And at a certain point, you can argue we might know more about these drugs and devices than they do, the ones who invented some of this stuff, because we just keep using it again and again. We have all the data before and after. So I think that's what gets folks excited, and that's what gets pharma biotechs excited about working with us to jointly come up with answers here. But the whole point is to enable all of you to do this as well. And just putting in a plug for one of our lab's papers here, Rohit Vaishis did this work on using just all the COVID data and all the antibody data, serology data that, people just get serology tests, but we also have the PCR tests. And we could show that, for example, no one, even after a positive test, we couldn't find more than 75, 80% of our own patients seroconverting after a positive COVID test. So we can learn a lot about COVID. We can learn about every single condition that we know and diagnose and treat in all of medicine with this data. And the we includes all of you with all of these tools here. The final slide here is how do these two relate to each other? We got UCSF data, which is deeper. We got the text, we got the images, of course, the structured data. And the UCY data is wider. It's wider, but we don't have the images. We're just figuring out the text right now. So if you want to go deeper, need images and text, get things started at UCSF. And then when you're ready, or if you could just work with the structured data, you scale across the other UCs. And the other UCs also have a similar type of setup. So everything we're learning from UCSF and the other campuses, we start to slowly move into the UC-wide repository. So I'm going to thank an incredible team, not just at the Baker Institute, but across all the different institutes and departments at UCSF that help us build the information commons for you. We build all of this for you. So I encourage you to really step in and learn how to do this, doing a research rotation or anything else. An incredible team across the University of California that makes all of this data possible for all of us and all of you UC-wide. So thank you.
That was fun. Thank you. Bring everybody up here. So let me start with a couple of questions, and we'll get some from the audience and online. I'm not sure which one should answer. Maybe Atul to start. How important is the volume here? The numbers at UCSF are big. The numbers in UC are bigger, but maybe 5x. When Cerner says we're going to aggregate all of, Oracle says we're going to aggregate all of Cerner's data to have a national database, that's bigger still. But at what point is there diminishing returns? Is the additional volume helpful statistically, or is it helpful in terms of the diversity of patients and settings? How do you think about volume?
Yeah, so that's an easy question. So volume is certainly important. Just in terms of sheer numbers, we're probably one of the larger repositories like this. But it's not just data. These are our patients. So we can also use them for cohort discovery to recruit them into trials. It's also easier for us to say that nothing is really rare in the University of California, right? A rare condition in a small hospital, even a single academic medical center, we probably see many of those across UC. My favorite example is a pharma company who was interested in how their drug was performing in patients that had a cancer genome sequence with a particular mutation, okay? And we had 7 of them at UC, right? That's 7 more than they could find anywhere else in the country. So it's not about 7 million. Sometimes it comes down to the right 7. But you need the volume to get to those right patients.
Any other comments? All right. The issue of case mix adjustment and confounding, maybe you can start, but anybody. All right, we find that patients who happen to get drug X did better than patients who got drug Y. There's a lot of things that might be different about patients who got drug X. In fact, maybe they were healthier, maybe their doctors are better, maybe who knows. So how do you look at that and say, I can actually make sense of an association and it's not all sorts of confounding?
Well, I think it depends on what question in particular you're asking. And certainly, like when you look at pregnancy, there's a vast difference across the United States. If you're being treated at UCSF, you're being treated in a community practice as to what you're getting. And so I think you have to look at hard endpoints that don't change. So things like birth defects and preterm birth and things that you can actually predict and that are objective outcomes.
Any other comments? Yeah.
Yeah, I think there's a science and that's developing here on how to use this data, right? We can't say that the book is already written. We're all kind of writing the book as it's happening. There's a science of propensity score matching, for example, right? We can try to ascertain what the physician's bias might have been in choosing one over another, but we can also use the rest of the record to try to compute a score to help match those. So I think that science and behind that methodology is certainly still to be developed. It's happening at UCSF.
Yeah.
Yeah, we've been using some of these approaches to first try and identify what those confounders are across institutions. We can do this at UCSF, we can do it UC-wide, we can look at Mount Sinai or Cerner or other data, and even outside the US, and see what these confounders might be, and then do various sensitivity analysis to see where it might make sense in terms of getting the population that's matched the best.
And then how do you deal with, how important is biologic plausibility, or you had a hunch for whatever reason that this drug was going to work, or this association was real? Because I can imagine a world where you're just pushing a button and it's dredging and it finds that this one thing is associated with this other thing, and it turns out to just be kind of a random association. So how important is it that you have a suspicion or there's biologic plausibility that something's real?
So from my work, what we've seen is if several lines of evidence align, maybe from molecular data and clinical data, if we see the same signals or same biological pathways coming up, then there's kind of more confidence. But again, I look at this as a way to generate hypotheses that then still need to be validated to better understand the mechanism.
Yeah, I think that from a clinician standpoint, I think that's a big problem, because there are some clinical decision support tools where you can plug in factor X, Y, and Z, and the answer that comes out makes absolutely no sense with respect to how you practice and the clinical trial data that you have. An example is one that was made for which biologic to pick for inflammatory bowel disease, and it had a patient who had fistulizing disease, and it said that vedolizumab is the best choice, when we know it doesn't work for it at all. So you have to be very careful and test multiple different hypotheses, but in the end, if it doesn't make sense from a biologic standpoint, I don't think we're going to believe it. And so that's where I would turn to all of you to say, well, how do you fix that?
Yeah, I'll add that by collaborating with individuals in Baker, I think a big take-home point is it's not machine versus human, it's a human and machine, right? And we're really talking about using our clinical domain expertise and leveraging that with the computational benefits of big data, and together trying to get to an answer, right? And when we think about trying to translate these findings into applications, being able to understand and explain what we found is really important. So, and I didn't show it in my data, but one of the big benefits and things that we have to think about when we choose specific models are how well can we understand the model and explain it to users, because that's going to be important when we think about how well it's going to be, the uptake is going to be, right?
Let's take a couple, if anybody here has one, but I think we have online.
Thank you so much. So a couple of questions on how do you all deal with missing data, and how do you all deal with the fidelity of the data with the busy clinician, maybe not necessarily putting in all the relevant ICD-10 codes?
You want to handle the missing one? I'll handle the fidelity.
Good.
All right, I'll do, so the missing data. So medicine is sparse, right? We don't measure everything on everyone, right? In every single encounter. So by definition, everything is missing, right? We have very few actual measurements. That being said, we might have anomalies. We might have, the lab tube, the tube that didn't make it to lab and things like that. So I think there are a lot of different ways to impute different values or just come up with statistical tests that don't require complete data, right? I think that's there. Regarding the fidelity of the data, that's usually the first question I get is why, is this messy data? And I don't believe this is messy data. I think medicine is a messy world, and EHRs capture that mess quite accurately. And so I really don't think it's messy data. But at the same time, let me put it this way. The narrative I usually give is, God forbid, if one of our doctors is hit by a bus and another doctor needs to come in to take over that care, everything that doctor needs to take over the care of that patient is in that record, right? There are no shadow anything anymore. So if a new doctor can start from scratch, pick up what the care should be for that patient, then it's up to us to teach the computer how to do that as well. And we're not there yet, but everything should be in that record to actually do it.
Just to add a point that's often made, which is, you want to look at messy data, look at what people put into Google search and how it figures out what you were thinking, because
we know how to
it's
care for
totally
patients.
messy, right?
Yeah.
But it's also the amount of data. Yes, the data is messy, but there's a lot of it. And we should be able to apply algorithms or develop algorithms to use it in a way to generate hypotheses or validate them. So we should figure out a way to do it, and that's what we're striving towards.
Last one is, how are you using these data to improve operations at UCSF or beyond?
So that's actually a great issue. We certainly use it. I can again go to inflammatory bowel disease. We use it for QI. So we see like this is what we should be doing. What are we not doing? And we can measure that. We use it for national and international guidelines to say to come to a consensus across the globe on how to manage this very complex patient population. And I think you do it for every single class is you use the data to find out what the outcomes are, what they should be, and what we're missing. And then you relay it to people, you teach them.
I could really spend an hour on how we've improved and, cost of care, for example. And my favorite example is IV acetaminophen, which we're not using as much here anymore because we have a master list of every single doc, nurse practitioner in the entire University of California who prescribes this drug. And it's about inappropriate use, right? Why does a patient get IV acetaminophen surrounded by other oral drugs? That's a complicated query. That's very easy to do with EHR data. When you have a master list of who's ordering it, it's almost like the Hawthorne effect. You'll look at this, it starts to improve by itself. And indeed, our use of IV acetaminophen, a very expensive drug we don't need to use, has started to plummet. That's one of many examples.
Let me ask one last question. A lot of what you all talked about is biopsying this data to get more insights for research purposes or maybe quality improvement purposes, sort of almost after the fact. When will the fantasy happen that I am taking care of a patient and the computer tells me in a way that's useful, patients like this did better on this other drug? Or, doctor, your diagnosis I think is wrong because you think it's X, but every patient that looks like this in our huge database turned out to have Y, or your patient isn't responding to therapy the way we would have expected. In real time, providing it to me as a frontline clinician to change my behavior in real time. Coming, not coming, 5 years, 10 years, and why is it so hard?
Come on, Bob, you asked that in the last question. We have 10 seconds to answer, right?
Go over.
So of course,
Take it 30
we're all
seconds.
going to be
Go ahead,
promoted.
go ahead,
So
go ahead.
I think it's coming, and you can argue about when. But right now, I think most physicians still view Apex or Epic as a documentation tool, right? It's not still there for decision support, though there are some decision support tools. You can argue about the success of some of them, like the sepsis predictors and others. But I think those are coming, right? The first 5 or 10 of these are going to fail, but they're coming. But we got to stop viewing these as just a documentation tool to start to look at Epic before we even go into the room, right? Or even the week before, the night before. We're not really in that mode yet as clinicians, but I think we got to get there and the tools will respond.
Great. Anyone to add? Yeah, go ahead.
I'll add to that. I think this is really important in something like a rare disease, right? Where providers who are caring for it may not have the numbers to have the pattern recognition that's necessary to make insightful diagnostic and management decisions. And so clinical decision support tools become crucial to translating evidence into practice for rare diseases. What we have realized is that the first thing we need to do is to make the EHR work for us, that it's no longer just a documentation tool, but it needs to have secondary purpose. And in order to do that, we need to streamline some of the ways that we're collecting data so that we can then repurpose it. So we can pull things like an age and BMI and your pulmonary function test and diagnosis and say, oh, this is someone who maybe we should be referring for transplant now as opposed to 10, a year from now. Yeah. That requires really thinking carefully about the fidelity of the data and how we can proactively improve it.
Okay, you'd want to make a last comment?
Yeah, I wanted to say, so I'm a computational scientist, I'm not a clinician, and I want to go back to something Erica mentioned. I think the way that this is going to move forward is if we start collaborating closer together so that I better understand as a computational scientist, what are the relevant clinical questions and what are the models that I'm building that can then help move the needle, whether it's therapeutics or diagnostics, and go back into the system. And I think that's really something that will help. I don't know when, but I'm hoping that we can get there soon.
Yeah. Anything you want to add?
Yeah, I think to what Erica was saying, I think we need to teach physicians within our own specialties to enter data in a systematic way that's standardized, because that's why things are missing. People just randomly enter data as opposed to, I think Erica showed a beautiful way of just picking the different things so that, for example, in our endoscopy now, we use a standard scoring system. So you can pull that out and everybody is scored for their Crohn's and ulcerative colitis the same way. And that it was easy to do and everyone did it. But I think we need it for all disease states so that the data makes sense and is usable.
Great. Fantastic session, totally cutting edge, exciting, frustrating, but we're clearly getting there and all of you are helping us get there. So thank you so much. Really appreciate it.
Thank you.